This method can help answer key questions in the material engineering field, such as the antimicrobial properties, which are very desirable for many bioengineering applications.
This technique can be utilized as a consistent protocol across laboratories to help less experienced researchers who require in-depth step-by-step procedures to follow for accurate results.
We first had the idea for this method when we started looking for antimicrobial protocols and realized there were no detailed protocols or visual in the literature.
In this study, 4 advanced materials with different chemical natures and a control material will be tested for antimicrobial activity. Prepare each material by cutting it into 10 mm diameter discs. Next, sterilize each specimen by immersion in 70% ethanol for 10 minutes, followed by ultraviolet radiation for 1 hour per side. Three different microorganisms will be used to test the antimicrobial activity of the advanced materials: the gram-positive bacteria Staphylococcus aureus, the gram-negative bacteria Escherichia coli, and the yeast Candida albicans. Using a sterile cotton swab, resuspend a few colonies of each microorganism in 25 milliliters of tryptic soy broth, or TSB, contained in a sterile centrifuge tube. Vortex for 1 minute to achieve uniform mixing. Measure the absorbance at 540 nanometers for each microorganism culture with a spectrophotometer. Adjust the concentration of each